Okay, so we continue with chapter 2. We are talking now about a different kind of cell division. In the previous recording, I talked about the cell cycle, which is the type of cell division that produces genetically identical cells for the purpose of organism growth, tissue repair, and maintenance. Now we're going to talk about sexual reproduction, which has a lot of importance in genetics, and we are going to focus on the chromosomal part of sexual reproduction, which is a big part of it. And what we're going to look at is, first of all, the overall of sexual reproduction. Sexual reproduction is the big umbrella topic, and there are two processes that make up sexual reproduction. First, meiosis, then fertilization. So we need to be very clear about that. Meiosis comes first in the sexual reproduction cycle. And meiosis is a cell division, yes, but it's not the same kind of cell division as the cell cycle. Meiosis makes haploid cells. It's a cell division that makes haploid cells. In male animals, these haploid cells are called sperm. In females, these are called eggs or ova. So that's only the first part of sexual reproduction. The good news is that most gametes, most eggs and sperm, die. And that's good news, because if every egg and every sperm were fertilized, then there would be way too many of every kind of organism on Earth. So that most gametes die. But occasionally you have an egg and a sperm that fuse together, and that's also important. That's the second part of sexual reproduction. And the fusion of those two haploid cells is called fertilization. And fertilization makes zygotes. Zygote is the fertilized egg. It is the first cell of the new individual. So this diagram here I found from on the genome.gov website. And I like it because it gives us a good image of what we mean by haploid and then what we mean by diploid. Ploidy is the word ploid, or the root word, or the root ploid means sets. And diploid means two sets. And the ploidy is a characteristic of the whole cell. In other words, if you have a whole cell, any kind of cell, the number of sets in the nucleus of that cell is called the ploidy of the cell. So a diploid cell, di means two, so diploid cells contain two sets of chromosomes. Now in humans, each set of chromosomes is 23 chromosomes of all different sizes. So here's a set of human chromosomes, and all these 23, you can see they're numbered 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, for example, all the way to 22, and then it looks like the Y chromosome. So there's 23 different size chromosomes that make up a set, and that would be what would be inside of the sperm. And also you'll notice that the chromosomes in a sperm are in the monad form, by the way. But the fact that there's one whole set of chromosomes, one of each different type of those 23 different chromosomes in one sperm, in any one sperm. In the same way, there are 23 chromosomes, one of each different type. All these, they're arranged in order by size, except for the X chromosome. And so 23 chromosomes in an egg. And that's what's inside the egg. So the egg is haploid. Or you could also use the term monoploid, meaning one set. So if a normal organism cells, the non-sex cells, are diploid, we're talking about muscle cells, blood cells, skin cells, brain cells. Those are the typical body cells. If those are diploid, meaning they contain two sets of chromosomes, then the term haploid just means half of the normal number of sets. So then haploid would be one set. So a sperm contains one full set of chromosomes, An egg contains one full set. And when the egg and sperm fuse together, not only do their cell membranes fuse together, but you can see here the process of the two nuclei fusing together. And eventually you end up with a single cell called the zygote with a single nucleus. And in the nucleus, there are two full sets of chromosomes spread out around the nucleus. Now, in this image, they arranged them where the two copies of chromosome 1 have been placed next to each other. The two separate copies of chromosome 2 have been placed right next to each other. But these are not sister chromatids. They're not connected to each other. They're just two copies that exist in a kind of a jumbled way in the same nucleus. They wouldn't be this organized in a real nucleus. But this is just showing you visually that there's two of each of these chromosomes in the nucleus of the zygote. So that gives us a sense of what sets are. So diploid just means two sets of chromosomes. So in humans, that the diploid number is 46. And so that means each of your cells has 46 chromosomes, except if you're a male, your eggs have 23. If you're female, if you're a male, your sperm have 23, sorry. And if you're a female, your eggs have 23. So that's what diploid and haploid are. But the point is that because meiosis produces cells that are haploid, then when two haploid cells fuse, the new individual returns to being diploid like each of the parents. All right, a brief review of meiosis. Meiosis 1 is called the reductional division because by the end of meiosis 1, the product cells, or sometimes called daughter cells, are haploid cells. So in this example, they show you a starting cell that has two long chromosomes and two medium-sized chromosomes. And the long chromosomes and the medium-sized chromosomes have already replicated, so you're seeing them each in their dyad form. But this would be one long chromosome in the dyad form. This would be the other long chromosome, also in the dyad form. And this would be a pair of chromosomes or a pair of homologous chromosomes. So you have a pair of long chromosomes and you have a pair of short chromosomes. And that they're lazy. They didn't want to, I guess, draw any more than that. But in humans, you would have 23 pairs of chromosomes in a diploid cell. Now, only in meiosis, though, do they actually come together like this. The two of the same type, the homologous pair, actually seeks each other out. And that's the only time in a cell's life. If a cell goes through meiosis, that's the only time when the homologous chromosomes seek each other out. And they pair up and they touch and they undergo something called synapsis and crossing over. And after that, which they're not really showing in this diagram, after synapsis and crossing over, the chromosomes align in their pairs at the midline or metaphase plate, cell divides. And so each of the product cells of the first division contains, you can see, one long chromosome and one medium chromosome, which in this example is a full set. The other cell contains one long and one medium. So by the end of the first division of meiosis, each cell is haploid. The second division is simpler. It's just the lining up of the dyad chromosomes individually at the midline of the cell, and the sister chromatids are pulled apart. And so each of the daughter cells of meiosis 2 contains the monad chromosomes here. And yet we still say that each cell has one long chromosome and one medium, so still a full set. So the product cells at the end of meiosis 1 are haploid, and the product cells at the end of meiosis 2 are haploid. However, the product cells at the end of meiosis 1 are haploid, but they contain dyad form chromosomes, whereas the product cell at the end of meiosis II is haploid but contains monad chromosomes. These cells at the end of meiosis II cannot go back and start over in meiosis I again. This is not a cycle. This cell division ends with these product cells that are haploid. And like I said before, in meiosis, these are gametes, eggs or sperm. Now, in any organism's body, you don't have one cell that goes through meiosis. You have hundreds for females or thousands and millions, I should say, for males. So this is just showing the concept of what happens each time meiosis happens. All right, so we're going to briefly go through, like we did before, the number of the chromosome form in each stage and the number of chromosomes per cell. So prior to prophase 1, there is a DNA replication similar to S phase of the cell cycle. And so in prophase 1 all the way through meiosis 1, the chromosomes are in the dyad form. However, in prophase 1, the number of chromosomes per cell is the diploid number. So in human, that would be 46. In metaphase 1, it's the diploid number. In anaphase 1, it's the diploid number. However, in anaphase 1, you would have one set of chromosomes moving towards the left end of the cell, the other set of chromosomes moving towards the right end of the cell. And by the end of cytokinesis, each of the product cells contains a haploid number of chromosomes. So going into prophase 2, the chromosomes are still in the dyad form, but the cell has a haploid number of chromosomes. In metaphase 2, the chromosomes are dyads and the cell still has a haploid number. In anaphase 2, this is where the dyads are pulled apart. So this is a transitional point. And so that haploid number, I guess you could say, briefly doubles to a diploid number, if you wanted to be technical. And then one full set of chromosomes moving to the left, one full set of chromosomes moving to the right. And by the end of cytokinesis after telophase 2, each cell would contain a haploid number of chromosomes. So there are some diagrams and some animations in your text that I would definitely direct you to really put this visually in your head if it's not there. All right, so we'll start the next lecture section with some problem solving.